Good day to everyone. So I'm Li Zhi Xuan. Now I will explain about the STPM Biology Semester 1 Chapter 2 Organelles topic. So this video will explain about the structure of endoplasmic reticulum and also its function. Okay, so as you guys know that there are two types of endoplasmic reticulum, which is SER and also RER. So smooth endoplasmic reticulum and also rough endoplasmic reticulum. So the general that you guys know the difference is RER, rough endoplasmic reticulum, is covered with ribosome. So when you see that the endoplasmic reticulum that containing ribosome, then it is the RER. And the smooth endoplasmic reticulum that does not have the place ER part that does not have the ribosome, you will consider as smooth endoplasmic reticulum, right? Okay, so normally if you want to explain the structure of endoplasmic reticulum, so you need to mention that endoplasmic reticulum normally is the network of tubule and membrane bound sets, which is called as cisterna, like this. This one is the scientific structure that you need to know. Network of tubule, this is a tubule structure, and it is a membrane-bound set, and we known as the cisterna. And these channels are continuous with outer layer of the nuclear envelope. So, RER and SER are surrounded with the nucleus one. This is a key structure to differentiate between Golgi apparatus and also endoplasmic reticulum. Because Golgi apparatus were not associated with the nucleus one. Only the ER. ER is associated with the nucleus. It is located at the side of the nucleus. Why? Later you will see it, the function. And then the ER membrane separate the internal compartment we call as cisterna space. So there are the space we call as cisterna space lah. And then RER and SER differences is the presence of ribosome. And in terms of the structure, also have difference. RER is the flattened tubule structure. It is the flat structure. But in SER, it is not a flat structure. It is the interconnected tubule structure. They are interconnected to each other one. You can see this. It is interconnected tubule in each other one. But you can see that RER is more like a paper. You can say like a paper. It is the flat structure. But SER is interconnected tubule structure, like a pipe one. It is look like a pipe. So it does not have the vibration to attach it lah. So this is the two differences that you need to know lah between RER and also SER. Okay, so the function for the RER, so normally the function of RER is protein synthesis, which is translation process will be occur at here. Normally translation process with the help of the ribosome to produce at here. So normally your polypeptide, meaning the protein will be formed, will enter the cisterna space and fold into its functional shape according to the structure lah, either in primary, secondary, tertiary, or quaternary structure. And then once they already form, you can see that they will part of the vesicle we call as secretory protein is formed and attached through the attachment of carbohydrate and polypeptide. And this secretory protein will be wrapped in the membrane of the vesicle, which is called as transitional endoplasmic reticulum. And this transitional endoplasmic reticulum will part off to form transport vesicle to transit to the Golgi apparatus. So meaning that once the protein is already formed in the ribosome, and then they will go to the cisterna space to form the necessary structure of the protein, either primary, secondary, tertiary, or quaternary. Once they already formed, they will fuse to the structure of the RER to form the transitional ER. I will form like this. Already got the circular shape meaning that the transitional ER containing secretory protein already. And then once transitional ER is formed, they will part off to form the transport vesicle. So this vesicle later will go down to the Golgi apparatus for the further modification. Okay, so this is function of RER. But function of SER is totally different between RER. RER normally is about the function of protein, the synthesis of protein and transport of the protein to the Golgi apparatus. But for function of SER is totally opposite, totally different. And SER is containing enzyme. They were containing enzyme that play the major role in lipid synthesis, which is a lipid anabolism to synthesize lipid one. So including your oil, your fat, your steroid, and also phospholipid. And normally this one is occur at your sex organ and also adrenal gland. And another one is also play the role in carbohydrate metabolism. And the third one is containing enzyme that can detoxification, play the role in detoxification for the poison, for the drug by adding the OH group to making them more soluble and easy to flush from body. So this one is occur in the liver. The liver will containing many SER to detoxify the drug and also the alcoholic drink. And another function is inside the muscle. When inside the muscle, they are also containing many SER, which is SERs, which is play the role in muscle contraction. When they play the role in muscle contraction, which they will pump the calcium ion from the cytosol to the ER lumen to trigger the contraction of the muscle cell. This one is later in semester 2, you will learn about the contraction mechanism of muscle contraction. One more detail about that. But for now, you just only know that SER, they will play the role in trigger of contraction of muscle by pump the calcium ion from the cytosol of the cell into the ER lumen of the cell. That's all you need to know. So there are 4 major functions that you need to know for inside the SER. So I think that's all for this video about the endoplasmic reticulum. So hopefully this video you can understand about the structure and also function of the endoplasmic reticulum. So thank you for watching. Bye.